Hello, thank you for coming. I'm Cottrell, and over the summer I used a repertoire analysis to investigate the effect of supplements on my immune system and my diabetic brother's immune system. Because of COVID-19, I was interested in trying to find healthy ways to boost the immune system so that it could better fight off illnesses. This is what led me to test the supplements known to positively affect certain illnesses for their ability to make the immune system more healthy and diverse. So, before I start my presentation, I would like to thank Mr. K for this opportunity. Also, thank you, Dr. Jianhan, Dr. Brennan, Dr. Pan, and Dr. Cottrell for all of your expertise and guidance. To begin, I'll introduce what in the immune system I look for to explain the health of my and my brother's immune system. So, specifically, I looked at the T cells in the immune system. Because T cells are part of the adaptive immunity of the immune system, T cells must detect a specific antigen of a pathogen to be activated. This means that T cells only defend against diseases when they find a disease that they're made for. T cells are an essential part of the immune system. They originate from progenitor cells in the bone marrow, and then they travel up the bloodstream to reach the thymus, which is the gland next to the heart, where they'll undergo a series of maturation steps. One of these steps is the rearrangement of the antigen receptor, which is responsible for detecting pathogens. If the T cell detects your own body, it dies in the thymus. The T cells that survive this selection become a part of the body's immune repertoire, which are all the T cells inside of your body. So T cells detect specific pathogens by interacting with the pathogen's antigens. And they do this with their T cell receptor, which is the shape that kind of looks like a Y in the middle of the page. Immune diversity is generated by T cells having different CDR3s, which is the gene that codes for what the T cell receptor recognizes. On the left of the photo, you will see the VDJ genes, which are under the heavy chain genes label. When making a CDR3 for the T cell, these genes are randomly selected to be put together. For disease prevention, the immune repertoire is a measure of wellness. On the left is a map of unique CDR3s for a diverse immune system, which is called a tree map. The D50 is a statistical measure of diversity, and the higher it is, the more healthy the immune system. On the right is another map of another tree map of unique CDR3s for an unhealthy individual. Here the diversity is low, and the large single color regions represent a single type of T cells that is present because of an ongoing condition or the memory of a prior illness. Low diversity is an indication of unhealthy immune system, while high diversity is an indication of a healthy immune system. So the general idea is that more diversity means that there are different T cells to recognize pathogens, and therefore the individual has a healthy immune system. Type 1 diabetes develops when the T cells that recognize that are supposed to detect foreign invaders actually recognize cells on top of your pancreas, which are vital for the production of insulin. This leads to the immune system to attack the pancreas. The results of type 1 diabetes include a high diversity of dysfunctional T cells that are less effective and may even recognize your own body. Type 1 diabetes also results in a lot of oxidative stress. In order to choose supplements that may potentially aid immune system function, I looked for biological processes that interact with the immune system. Oxidative stress is the chemical imbalance of reactive oxygen and reactive nitrogen species. High level of ROS, or reactive oxygen species, have been shown to negatively impact immune system function. ROS are free radicals, and free radicals are created by normal cellular processes. And as I said earlier, type 1 diabetes increases oxidative stress. The way the body controls the amount of ROS are antioxidants. Antioxidants donate an electron to a free radical or reactive molecule and then convert it to a stable form. In this way, antioxidants can lower oxidative stress by neutralizing ROS. The endocannabinoid system is found throughout the body. Two of its receptors, CB1 and CB2, are embedded in the membrane of cells throughout the body. CB2 receptors are primarily found in peripheral organs and the cells of the immune system. The activation of CB2 receptors is involved in immune modulation. The formation of T cell subsets, so this is memory helper T cells and natural killer T cells, require the endocannabinoid receptor CB2. And also, mice deficient in CB2 were found to have profound deficiency in memory CD4 cells. The first supplement, NAC, was chosen to lower oxidative stress by increasing the level of the antioxidant glutathione, or GSH. So NAC is an acetylated precursor of the amino acid L-cysteine, and L-cysteine travels, or NAC travels into your body, and then in the liver, it leaves L-cysteine, and L-cysteine is a necessary reactant in the synthesis of GSH. Taking NAC will increase glutathione levels, which is a key natural antioxidant and thus acts on oxidative stress. The NAC used was pharmaceutical grade and sourced from Nutravita. And the second supplement was chosen to interact with the endocannabinoid system. Noni has been used in traditional Polynesian medicine for centuries to treat a variety of illnesses. Research has shown that Noni binds to the CB2 receptor in the endocannabinoid system, and it's listed as an immune system enhancer, anti-inflammatory, anti-cancer agent, and type 2 glucose level modulator. From there, research questions and an experimental protocol was developed. So my research questions are, will an antioxidant amino acid, NAC, that produces glutathione cause an increase in immune system diversity? Will any observed changes in immune system diversity be different between the non and type 1 diabetic siblings? Will a traditional medicine, Noni, that is an endocannabinoid or cannabinometic, cause a change in immune system diversity? And will there be any determinable synergistic effects when both compounds are used at once? A protocol was established to address the experimental questions. And so each supplement, NAC and Noni, were to be taken for 1 week, and this was followed by a 2-week clean-out period. The chosen sequence was NAC, clean-out, Noni, clean-out, and then a combination of both. Dosages were taken from established research and vendor recommendations. So 1,200 milligrams of NAC was taken twice a day at 9 a.m. and 6:30 p.m., and 1 ounce of Noni was taken twice a day at 9 a.m. and 6:30 p.m. Blood samples were drawn always at 9:30 a.m. for the baseline, 1-day and 5-day readings. So how does iRepertoire get the immune repertoire? And how do you send in your data so that you can get out how many T cells and your diversity of the immune system? And so first, iRepertoire will send you a kit, and you have to take a blood sample from a finger prick. And so you see on the left, it says peripheral blood sample. You have to fill in all of the little divots. And once you send it back to iRepertoire to analyze, they use reverse transcriptase to convert RNA into DNA. And then they also use multiplex PCR and next-gen sequencing to create tree maps and other good analysis tools. And so using the data collected from iRepertoire, I addressed my research questions. And so the raw iRepertoire data is a list of frequency of occurrences for each unique T cell CDR3 beta receptor. And so because the samples were taken from a blood sample, and so the absolute number of T cells is not consistent because the amount of blood that you put into one will not be the same for each of the little divots, as well as tests. The number of T cells is not consistent from sample to sample. However, the distinct number of CDR3 is a measure of diversity from sample to sample. On the left is the total number of CDR3 in the blood sample collected, and on the bottom are the trials. My brother's data is on the blue line, and my data is the orange line. In the NAC trial, I had a steady increase in my distinct CDR3, which indicates that my immune system got healthier, while my brother's distinct CDR3 went down. Both of us followed the same trend during the 2-week cleanout period, and we had similar baselines after NAC. Although my brother's data for day one, the day one Noni, was bad, we see a very similar trend for both me and my brother, in which distinct CDR3 went down after 5 days of taking Noni. Nothing much happened on the first day as opposed to NAC, where there was a large difference in my immune repertoire and my brother's immune repertoire. The cleanout period after Noni also brought both me, my brother, and I to relatively the same place in immune system diversity. For the NAC and Noni trial, traces of NAC and Noni results are present. The day one was a large change, like in the NAC trial. However, both of us went down after 5 days, which is believed to be a trend for Noni. However, overall, if we look at the D50 as well as the diversity index, it seems that NAC that produces GSH does not diversify the immune repertoire when it is taken over time. So the participants, me and my brother, had a very large opposite reaction after 1 day. And then both participants returned very close to the baseline after 5 days of NAC. For my second research question, to see any observed changes in the immune system diversity, and if they were different between me and my diabetic brother, there is a very distinct difference in all of my brother's tests and mine. So can you tell who is who? You can, because on the diversity map on the left shows a large block of a single clonotype, which is in the top right of the tree map, and it's a dark green blob. This would be expected for a type 1 diabetic, as they express a large number of T cells that will attack the pancreas. And this is activated by, then it's activated by recognizing antigens expressed by the islet cells on the pancreas. Another way to distinguish me and my brother's tests were that CDR3 that were associated with type 1 beta cell epitopes, which means that this CDR3 will recognize my brother's islet cells on the pancreas, and that means that they're activated by it, and that means that there will be immune response against it, which is also how we see the large green blob, so that it's a byproduct of him being recognizing his own system. However, there are a small number that showed up with some frequency between participants. And this CDR3 also did not show up in any of the data for me, and it was unique to my brother. So my research question number 3 was, will a traditional medicine Noni cause a change in the immune system diversity? And what was found was that Noni consistently decreased immune system diversity between me and my brother. The Shannon equitability index shows that both participants moved about 6% away from a perfect diverse distribution, which would have only 1 T cell per different T cell types found in the blood over taking 5 days of NAC. And this data also shows an independence of individual condition for the effect. So this means that the diabetic reacted the same way as I did. So it seems that it doesn't matter whether the participant is immune compromised or not for the Noni to decrease immune system diversity. And after a 2-week cleanout period, both participants saw the same diversity increase rebound effect. And so this is from point Noni 5. So that's right at the very end of these 3 dots, and that's all the way up to the baseline 3, so that's B3. Within the Noni data, there was a trend that correlated to the Noni response, and this is that both me and my brother had a similar change in frequency of the occurrence of our top 50 CDR3. It shows that a decrease in immune system diversity from Noni consumption correlates with an increase of the frequency of the top 50 CDR3s. So this means that there are more memory T cells in both my and my brother's body, and therefore the immune system diversity went down. The one on the left is my brother's data. The blue line is baseline before the Noni, and the orange line is after 5 days of Noni. The gray line is 2 weeks after stopping Noni. And on the right, this is me. The blue and orange data overlap and are the baseline before Noni and after 1 day of taking NAC. The gray data is 5 days of Noni. Yellow data is 2 weeks after stopping Noni. And both participants, the trend is the same and appears to be independent of individual conditions. The final question is if there are any observed synergy between the supplements, and the data does not support a synergistic effect between NAC and noni. However, when taken in combination, the data shows an effect over 5 days that's similar to noni, but somewhat dampened by the addition of NAC. So the overall decrease in diversity was smaller, but the same for both participants just with the pure noni test. So for my conclusions, the immune repertoire could be used as a diagnostic. It detects unique and shared clonotypes, and it is scientifically possible to establish that a CDR3 is associated with an autoimmune disorder. The immune repertoire can be changed by taking certain supplements, and the mechanism by which the supplements interact with the immune system can cause an individual effect or a common effect, like we see with noni. Increasing immune repertoire contributes to the general wellness with regard to fighting off pathogens, and inflammation-based conditions are known to be helped by noni. And this data supports a modulation in immune repertoire to decrease an overall diversity. And so this calms down my brother's immune system, and this could be a positive because it calms down all of the ineffective as well as the potentially self-harming T cells in his system. And so what I tell people if they're gonna, when they follow what I did is be in touch with your mentor, especially during the research phase of the project. For me, that was most of the project. So it would help more if I contacted my mentors more. However, don't overly stress about the project. Just do the work and try not to procrastinate, as well as don't be afraid of bad or unexpected data. I wanted to see what would increase immune diversity. However, what ended up happening was that I saw a similar trend for noni, and I thought that maybe perhaps this could help autoimmune disorders calm down their overreactive or their overactive immune system. Make the project into something of your own, and if I could do it again, I would like to increase the sample size, as this data is not relative to every person. So thank you again to Dr. Han, Dr. Pan, Dr. Oy, and Dr. Chan. These are my references.